Norman Maiwashe. I am with the Agricultural Research Council and responsible for managing R&D on animal production. And thank you very much for making the time to attend this session. I was reliably informed that this is going to be a very exciting session. My only worry was that this is 2 o'clock. 2 o'clock can be very tricky. But let me assure you that I have a very capable panel here that will make sure that you don't fall asleep. We have promised you that we're going to share some thoughts around the omics. And often these words are thrown around and we sometimes do not know where they come from. Who first started saying omics? And I think the omics word is very new in the scientific community. But the economists have been doing this for a long time. They've always had As a scientist, we feel that we have discovered something new. It's not that new. When I was reading on the subject, it was interesting to find that in the science space, the omics and genomics specifically, these are words that have been coined not long time ago, less than 40 years ago. That's when people started talking about genomics. And then later on in the 90s about phenomics. And if you do your Google search, you realize that there are more omics than we care to remember. So omics is very, very important. But what are these omics from the science perspective? This is nothing else other than us being able to look at biology and gain a much broader insights about the biology. So there's massive data that is coming through. But equally, this data also has noise in it. How are we going to separate signal from noise? And I think our colleagues here are going to share some thoughts around this. But we're not talking about omics because we just want to talk about things. We're talking about omics because something big is going to happen in 2050. We'll be close to 10 billion people inhabiting Earth. That's a huge number. But the challenge that we face is that the natural resources are dwindling. We have emerging threats. And those of you who were already born in 2019, 2020, you would remember that we had COVID. So it's not unthinkable that we'll be confronted with this kind of situations. I just had to make sure that you understand the task that we are faced with. So I'm not going to waste a lot of time. I'm going to introduce our panel. But before I do so, I just want to set the scene so that you can have an idea of what we have in our portfolio. So we have a full menu. Some of the things that they're going to talk about, they're going to talk about the lessons learned, the teething problems with these omics. How are we experiencing them? How are we overcoming them? And the other thing that they will look at is to say, how are these omics adding value? As society, we are less important on those things that do not add value. We don't want to be entertained. We have enough entertainment on its own. And the other point is around policy. Is the regulatory framework ready to embrace the omics? So they will talk about this. But of critical importance is that with all the industrial revolutions that we have witnessed, we have always made sure that we leave some people behind. How do we change things so that this time around, nobody gets left behind? So that is just meant for you to understand that it's going to be really exciting. Let me first introduce the first panelist. I'm going to give them 5 minutes each just to break the ice. And then we'll go on a marathon for 40 minutes. And we will leave 20 minutes for you as audience to ask questions. But if somewhere along the line we realize that we have spoken is enough, we might open the questions sooner. So if you just keep on showing that you are entertained, I may open the questions very soon. So there's no sleeping for you today. Let me introduce the first panelist, who is none other than Dr. Jasper Rees. Those who know Jasper, they know that he's a big fan of technology, biotechnology, and the omics. And he didn't get the gray hair from not speaking enough about this. He has spoken enough. That is where the gray hair comes from. Jasper was educated at Oxford University, and he also did his postdoc in the USA. He was a Wellcome Trust Senior Research Fellow at Oxford, and then a professor of biotechnology at the University of Western Cape. And this was long time ago in 1995. And from there, for the following 15 years, he played an important role in building one of the biggest biotechnology departments in the country. Jasper has a very long CV that is very decorated. In 2010, he moved to the ARC to establish what is now called the ARC Biotechnology Platform. In 2014, he rose through the ranks to become a group executive for research and innovation systems at the ARC. And from there, in 2020, Jasper joined the Botswana University of Agriculture and Natural Resources as its first vice chancellor. And from there, he implemented a strategy that brought the university to the level at which it is. Since 2020, Jasper is an extraordinary professor at the University of South Africa. Jasper, I'm going to give you 5 minutes. I'm going to time you to provide an opening statement.
Thank you, Norman, and welcome to everybody for this session on omics for food. And I need to reiterate that you're not going to eat the omics is going to eat you. So I think before COVID, it would be fair to say, how many of you knew what PCR was before COVID? No, it's okay if you did. Let's get a sense of the audience. How many of you knew what PCR was before COVID? And how many of you done PCR before COVID? Okay. So after COVID, how many people knew what PCR was? Everybody. So we had this enormous transformation. Everybody knew what PCR was. Actually, in reality, it was just a test. It was just something that we'd all become familiar with. And I went to Botswana in early 2020, and COVID came in almost immediately after. It was not my fault. But I had to close the university within 6 weeks of arriving, which was definitely my fault. And by the time we reopened, everybody was having PCR tests and antigen tests, and the whole community seemed to know what the difference was between PCR and antigen tests. And at the same time, when I joined the university in Botswana, I couldn't find anybody who could do a PCR. And by the time COVID had finished, I could get a PCR test in almost any shopping mall that I went to in Botswana, including one right up in the north in Kasane, where we found a little PCR lab at the back end of some facility. So these technologies come, and they can come very quickly. So when we look at what omics technologies are bringing us, I first sequenced DNA in 1981, 82, which is longer ago than most of you can remember. And I wasn't in a very advanced primary school. I was in university by then, so I'm ancient these days. And we didn't call it genomics. We were biochemists. And DNA technology was a biochemistry. And the people who trained me, for them, it was organic chemistry. And they treated DNA as an organic polymer, and everything we did was organic solvents and so on and so on. So very rapidly over the years, by another 10 years went by and genomics had started to become a word. And today we have genomics and proteomics and metabolomics and phenomics, and you can make up an omics, economics, that's what we're going to call it now. And these so what defines these technologies is not what the technology is, but that the technology is high throughput and generates huge volumes of data. And this is really transformative in terms of our understanding of a whole range of aspects through the food system. And at the same time, it's a technology. So when we look at questions, for example, of what do we do about policy, the answer is that it doesn't matter what the technology is. If you're testing for food safety, 30 years ago, you'd have spread stuff on a plate and seen what microbes grew and what. And today, we could look at it by DNA sequencing, we could look at it by proteomics, we could look at it by any metabolomics, we could look at food safety issues through a whole range of different high throughput technologies. And so the question becomes, can we implement these analytical techniques in the food chain? And I'm talking about from the chicken to the polony. Perhaps I shouldn't say polony because it sounds as if I'd be identifying a particular company with a particular problem with a particular microorganism. But if we're looking at the food system, is it practical for us to say we're going to be able to use omics technologies to test all the steps in that process in factories with multiple different product lines all over the country? And if we are, how does that relate to our current food safety regulatory systems, and do we need to change them in order to incorporate the use of these new technologies? And I would suggest we don't need to change our regulatory environment in order to address how we're going to introduce new technologies, any more than we would have done when we introduced PCR or mass spectroscopy or any of the other techniques that are important within that spectrum. So from a policy point of view, I think we have a strong regulatory system for food safety. Now, of course, the food system has many other aspects, and I'm not going to talk about plant breeding because Glendon will talk about plant breeding in a second. He'll talk about how omics technologies are impacting on that aspect of the food system. But I think that from a social point of view, we should see this as part of an evolving continuum of technologies that have developed and will continue to develop. And what's important is that they will continue to get cheaper, because the cost of doing all the different omics technologies, but DNA sequencing is a really good example. The cost of sequencing DNA has dropped dramatically in the last 10 years. And that reduction in cost, together with an increase in our ability to analyze many, many samples simultaneously, means that it's actually practical to look at how we would use these kinds of technologies to analyze and monitor food production systems and food safety. Norman, I'm going to stop at that point because there's lots more to talk about, but it will come in due course.
Thank you, Jasper, for the brilliant opening statement. I had timed you, and you stuck to 5 minutes. Let me then introduce the next speaker, who is Dr. Glendon Ashkoff. Glendon obtained his undergraduate degree in genetics and molecular biology, and also his PhD from the University of KZN. He began working for Panaseed in Greytown, and was later transferred to Stark Ayres, which is a vegetable seed company in Johannesburg in 2012. He is responsible for setting up the biotechnology lab, and now oversees all vegetable breeding, plant pathology, and biotech research tracks as a research director. Over to you, Glendon.
Um, I guess the angle I'd like to take this afternoon is where are we as a country, as a continent, with using these different omics technologies? And I'd like to kind of paint a picture of the company I work for, Stark Airs, and how it's developed for us over the last 15 years or so. I think the overall kind of overarching idea for me would be that science and research is a journey. I love Einstein's quote. He said, "Research is what I'm doing when I don't know what I'm doing." And it's that journey of learning things that I've been privileged to enjoy at Stark Airs. When I joined the company about 15 years ago, we had a small team of breeders. There were about 6 plant breeders scattered around the country. And only 2 of those 6 breeders were using marker-assisted selection. They were probably submitting in total 2 or 3 plates of leaf samples, sending it overseas to a lab in the US to do a couple of disease resistance markers for one of our crops. Within a year of being there, we'd actually started our own biotechnology lab. And within that first year, we managed to get most of our crops onto a really simple SSR-based marker-assisted selection platform, doing basic fingerprinting and basic MAS across many of our crops. A year later, we were able to convince our shareholders to invest in a high-throughput SNP genotyping platform, and we were the first in Africa to have any such high-throughput equipment. And what that really did for us, within the next 2 years, we were able to get all of our crops to generating multiple hundreds of data points, per chromosome. And as Jasper said, generating multiple amounts of data that we could use for fingerprinting, marker-assisted selection, QTL mapping, etc. And at that point, we felt like we were starting to understand the genetics, if that makes sense. 2 years later, we employed our first bioinformatician. Again, a big turning point for us. We started sequencing our parent lines, our breeding material, and we outsourced that sequencing, some to the ARC in the early days when Jasper was there, some to other companies overseas, but also locally. And as we started to understand the sequence data, we kind of added to our layer of genetic understanding a genomic layer of understanding that sequence data, telomere to telomere sequence data, helps us better make selections, better track our traits, helps our breeder understand their germplasm a whole lot better. And today we're at a point where all 14 of our breeders are generating between them 4 million data points out of our lab, processing about 120,000 samples through our own lab. And it's been an incredible transformation in the last 15 years. And so depending on where you're at, you might be in any one of those spectrums. We're still on a journey of learning and discovering. Some of the things that we'd love to do more, certainly it's happening in the field crops, but just starting in the veg crops, genomic selection, where we're able to track quantitative traits using markers across the genome to select much more accurately. We're exploring the area of pangenomics, doing long read sequencing on our breeding material, because the more we learn about the genomics, the more we realize that a third of the genome is possibly accessory genome. And often in those accessory components are unique traits that are helpful for breeding. And in terms of our contribution to the economy is generating vegetable varieties that farmers can grow, small-scale farmers can, subsist off and make a living, and the home gardener can also, take part. And so for us, it's been this kind of, even though we're in the corporate and we have a strong focus on R&D, we had to demonstrate kind of a stepwise introduction of these technologies. First, a DNA marker lab, tick, it went okay. Then a high-throughput system, tick. Then we added bioinformatics and genome sequencing tick. We weren't able to, in one step, combine a whole bunch of experts and a whole bunch of technology in one go. We've had to do it in a stepwise manner. And I think the result of that is that we've been able to learn significant, what's the word? Well, you learn by bumping your head. You learn by failing and making mistakes. We've had lots of learnings as we've gone along. And so I guess that would be my kind of opening statement.
Thank you very much, Glendon, for painting that picture. I think now we have a sense of the amount of work that is required to get off the ground. Last but not least, in terms of our panelists, she's bringing the light to our panel, being a female and us having celebrated the Women's Month. We're happy to have Dr. Kanyisile Hadebe, who is a senior researcher in genomics at the ARC Biotechnology Platform. Her research is focused on the characterization of biodiversity of indigenous animal genetic resources, with special interest on population relatedness and structure, signature of selection and adaptation, as well as environmental fitness. Her research application is in the development and optimization of genomics and digital tools for breeding and conservation program, specifically focusing on locally adapted breeds. She's involved in various community projects that assist village-based smallholder livestock farmers to improve record keeping so that they can manage their stocks better. She's a practicing natural scientist and has published over 20 peer-reviewed publications in local and international journals, and she's also rated by the NRF. Kanye, take it away.
I think I'll fade to my seat. Thank you so much for that introduction, Prof. So Omics research, it provides a state-of-art technology for us to explore for food quality, and also has contribution in development. But it comes in two ways. It can either benefit or it can also harm. So my line of work and the exposure that I've had is that, yes, it has benefited over the years, more privileged communities, but then we are leaving behind the less privileged communities. But with that being said, there's a thin line, like I said, between it's benefiting the communities and also it can harm the rural-based farming communities. And it's based on the practices of the lack of concerns, the lack of consideration for the cultural aspects of the farmers that we are improving. It also, the lack of or low consideration of and exploitation of the biological samples. So we collect samples from these farmers, but we never give back the results. Even when we bring back the results, it's nothing that they can use for their production improvement. So it leaves us more at the benefits, because we can create and have patents and have products, whereas there's low bringing back to the farmers or plowing back to the farmers with what the research that we've got. So that's where majority of the things that are very controversial to the application of omics in research, either in plants or in animals. So with that being said, when I design a study, I always think about one of the key things that the exposure that I have. I'm a goats specialist. I've been working with goats ever since. So with results recommendation that came in the 1990s, we found Boer goats being an improvement strategy for the goats industry, especially in the rural areas where Boer goats were donated to rural communities to improve their livestock. So with that, then became a trade-off of the adaptability. So we lost a lot of our adaptability to the production. So with that, I would like to trigger your thoughts to say, when do you stop the line? Should we really leave them behind because we don't consider their cultural, because that's the biggest thing that we lost. We lost the heritage of those animals, because now we've crossed them out. Where they had adaptable traits, now we've crossed them out and we're facing with climate change. So what do we do? Do we back cross them or do we leave them? Thank you so much.
Thank you very much, Kanye, for those thought-provoking ideas. I think you are quite spot on that omics on its own will not just change things. It's people and in terms of how this is used. Jasper, if I can just follow up with you, you indicated that for us to be able to do this properly, there are issues around regulatory frameworks to enable us to optimize the use of these omics. And you specifically indicated that in terms of food safety regulations, we are fine, we don't need to be changing anything. Will I be correct to say our regulatory framework is in order for us to proceed, or that will be simplistic?
So I think we have to separate the. I think we have to separate the use of these technologies from how we're going to apply them. So a number of years ago, a study was published by some colleagues in Stellenbosch, and they had looked at the identity of meat products in the food industry. And I think they looked at about 130 samples, and I think they claimed to have determined the presence of donkey in about 120 of the 130 samples. I'm probably exaggerating, it was probably 110, but it was a very high percentage. And they did that study without a proper regulatory framework. The samples weren't collected with appropriate consent, and the information leaked out into the community that one of the samples came from a halal butcher's in Durban. And the press got hold of this, and they sent the reporters off, and somebody leaked that the Durban, these butchers were in a particular street. So they went, and they found 2 halal butchers in the street. So they went to the first guy, and they said, "Was it you?" And he was smart, so he said, "No, it wasn't me. So they went to the second guy, who said, "I think it was me." But he said, "I'm a halal butcher." They'd been a halal family butcher for 50 years, deeply embedded in the community, and suddenly they were accused of having, wait for it, porcine DNA in their halal sausages. So this literally bankrupted this company, because suddenly the Muslim community didn't trust that it actually was halal, although it was certified by the Muslim Judicial Council. But there are 3 halal certification authorities in the country, and it was one of them. So they came to us and said, "Can you explore what's really going on?" And to cut a long story short, because this was already a long story, the meat in the sausages was what it should have been. It was lamb and beef. But the casing of the sausage, which had come from a factory in India, which had been certified as halal, was 20% porcine DNA and about 80% something else. So you don't have a regulatory system that addresses properly how you deal with that kind of information. But I think that we have to look at, so that's a level of information that was so much more precise. And yet the Halal Certification Authority said to us, "We've been to that factory. We guarantee it doesn't contain porcine products." But fortunately, their scientific advisors were people I had trained, and that's why they'd asked me to do the analysis. And they said, "Well, we believe you, but it means we've got to go back and review how we deal with our certification process, because you go and certify one day, and then 2 years later you assume everything's how it should be." And I think that when we look at our regulatory system, it isn't about omics technologies. It's about a robust testing and traceability and monitoring systems to ensure that what you thought you bought was what it should be, and was it safe, because food safety and food identity are 2 critical parts of the same process. And I don't think it's about should we regulate omics technologies for food safety and food identity. I think it's we have to be able to regulate food safety and identity, whatever technology we apply to the traceability and the safety aspects of the food system. And much as I love genomics and I'd love everything to be sequenced that I eat, I have to be practical and say that isn't achievable. And so the question becomes, at what level do we test and what level do we trace? And what does the rest of the world do? Which is another question.
Okay, thank you very much, Jasper, for expanding on this point. with any technology, there's always a resistance from society, because they need lots and lots of data to have comfort that whatever they're eating, it's pure and close to nature. Glendon, from your side, you have used marker-assisted selection. There are other ways of breeding. But generally, the public is wary of us sneaking something through the breeding program in terms of improving traits of economic importance. From your perspective, are you picking up that there is resistance from society in terms of this particular line has been bred through genomics and therefore we are skeptical?
Not specifically bred through genomics, but as a company where we sell to commercial farmers who I think have some measure of understanding of agriculture and science as a background, and we have a retail outlet, we sell small seed packets to the hobby market. We obviously like to be customer facing, and so we have a Facebook page for our company, and most of the comments and questions that come through those channels are from retailers, just the average housewife, person at home who's growing their seeds. And we've had questions like, how dare you be GMO, Stark Airs, because you're selling F1 hybrids? And simply misinformation, misunderstanding that a hybrid or an F1, which is a combination of two genetically different but stable and pure breeding parent lines of the same crop, constitutes a GMO. There's no understanding of that. And so we don't do any GMO. We're not in that line, and yet the public has a complete misperception of it. On some level, I think that plant breeding is genetic modification, because when our tomato breeder crosses two breeding lines, there's 30,000 genes on each side, and they don't know what they do, most of those genes. Whereas genetic modification in a lab, you're playing with one trait which you know exactly how it's controlled. So the level of precision and understanding from a laboratory GMO point of view, I think is much more tolerable for me as a consumer. The scientist knows what they're tinkering with. The breeder just chucks 30,000 genes on each side and hopes to come out. But I don't know if there should be resistance. This morning, part of my breakfast, I had fruit salad and yogurt. And there in the fruit salad was kiwi fruit, but it was not green. It was yellow, but it was not bleached. It was a GMO because a breeder had somehow taken the color gene, and presumably there's variation within kiwi fruit germplasm to allow that. And I think the real challenge that we have as a plant breeding community is we stand on the edge of a revolution of gene editing, not the conventional GMOs, agrobacterium and gene gun and biolistics, but gene editing with techniques like CRISPR-Cas and other base editors and precision editing where there's no insertion of foreign DNA, and our regulatory system is not able to cope. They can't identify between the two, although they're very different scientifically. So yes, we have an uphill battle, but we're on the cusp, and yet there's some challenges there.
Okay, we will come back to this. I think the audience may bring this back. I want us to look at the other side of the coin. Kanye, you spoke about your love for nature, genetic diversity must be preserved, and all this and that. And you specifically indicated that there are pros and cons of this technology. I mean, from a village farmer perspective, how should they be excited about this omics?
Thank you, Prof. So for my side, it needs to be practical. It needs to come down from omics to the farmer and to the animal. So if my interest or my breeding objectives as a farmer in Nkandla is coat color, my market is coat color, or it's adaptation because I'm in Limpopo in Musina where it's blazing hot. So even the responsibility of the researchers, the omic researchers, has to come in the cultural aspect. It has to cover the objectives of the farmers. And if you have them parts of the decision making, or at least in the advisory when we are continuing to do the research, then it allows them to know that they've contributed, and this is how they are going to fit into the programs and also in the improvements of the animals. Because sometimes we bring solutions that farmers don't need, or solutions that dilute or can erode the current, the adaptable populations. And then they are faced with more diseases, and then they have to now invest more in their livestock than they used to. So there's a thin line, or a rather a trending that we have to be careful of when we are bringing omics or these solutions into the communities. So for a farmer to be more comfortable, so in my experience, is when you work through with them. So you understand their dynamics. You understand if there's a horsey there, if it's communal land, you treat communal land different from a farmer who's established or could be emerging, but they've got their own resources. You know, there's a lot of people who are dependent on grants and these animals. So if you interfere with those animals, then you are messing up with their livelihoods. So that's the one thing that I really want us to be in consent of when we are working with communities, especially in this breeding, through omics research.
Thank you, Kanye. So the message is that we have to be context specific. Let me, as we conclude, because I think our audience are itching to ask questions and also make comments. This omics, I mean, this is not new in terms of big things happening. And the manner in which we have been able to harness this has differed. I just want maybe each one of you as a last thought before we go back to our audience. How important are partnerships in getting this to work? The public-private partnerships and so on. Jasper?
Well, I think they're critical. I think if we go back to the listeria outbreak, it was public laboratories which generated the data that allowed Tiger Brands to trace listeria back into the factories and determine where it came from. NICD and ARC, I'd understand, were responsible for that analysis, and the company wasn't in a position to do it. They don't have the facilities. They don't have the laboratories, don't have the expertise, as I understand it. And if I'm wrong, I'm happy to be wrong. But so it was a public, it was a public service that addressed that issue and got it under control. So the investment that we make as government organizations in these technologies allow them to be established in advance of companies necessarily feeling that they're ready to come in and make that kind of investment. And Glendon, I think, with Starcares is a really clear example of not just the technology process, but of being able to reach out to the ARC, as you did as early as 2012, where we had sequencing technology that was publicly funded, massively expensive, and absolutely appropriate for what Glendon was trying to do, which was, by anybody's standards, a pretty daring plan. And he came to us and said, we want to sequence all the species we work on. It was a bit daunting, actually, but it was done. But if he'd gone to his shareholders and said, I want a 15 million rand DNA sequencer to put as the first piece of equipment in my new lab, well, they would have said no, wouldn't they? So I think, it's about being able to create an ecosystem in which we have the right technologies in the country, we have the right expertise to use them. The demonstration of their use isn't cost effective in the beginning. It's brutally expensive, and if the public funding doesn't achieve that, it's unlikely that the private sector is going to be able to get on board and get these technologies to work. And then when they become cheaper, and you can get a PCR in every supermarket complex in the country, then it's become a practical application of omics technologies. But inevitably, it starts at the high end. It's crazy prices, and that is the nature of publicly funded research and for publicly funded facilities. If we're successful, we drive it out into the system, which might be hospitals, it might be factories, it might be companies doing breeding or whatever. And eventually, you'd like to have point-of-care diagnostics. So we say, you can test your polony and see if it's got listeria before you make your lunch. But we're not at that point yet. But when we are, people would just say, oh, it's normal, I tested my lunch and my lunch is safe to eat, so I'm going to eat it.
Yeah, so I think there are, we should actually be like in an ecosystem, one with multidisciplinary projects or any program or any initiative that comes, when it's got multi-stakeholders and expertise, the context of how I may think like a landscape genetics person, someone may think like a plant pathology, but we're just looking at the goats and where it's sitting or where it's based. So someone may look at it in a perspective of pasture. I'm looking at it in a perspective of genetics. Someone is looking at it in the perspective of health. So the person says, no, this animal is resistant to so and so we can place it here. As a landscape geneticist, I'm like, although it has the potential, but it's not a good production system because of the farming structure, if it's like communal or the resources that are available. Where someone from plants will be like, from pasture, can look at it to say, we've got enough feed to support the animal. So really, this animal is protected by multidisciplinary. So when it comes to the public sector, because of the way our structure of funding and is, sometimes we reach a point of we can't go any further because of not regulations, but there's only so much that we can do. But there's the pioneers within the industry, then they can take up on some of the things. So looking at the private end, we really need to work together. And I'll emphasize again, the inclusion of farmers, the inclusion of the extension workers or the extension services with from the bottom up. Sometimes you realize that the farmers there, or they already, when you see them being challenged, but they have been farming for years, and they've got enough knowledge, indigenous knowledge about these animals. And sometimes it even matches of what you say or what you see. But then because they've been farming, they actually know. You just need to buffer them in making the right decisions and keeping records, obviously for traceability but they know Jamludi, they know Uhlanya, they know all those animals and their pedigrees, So that's what I have to say.
Yeah, Kanye, you're very passionate about communal farmers. I hope
Owned business, we can't do everything we want to in-house. We can't, as Jasper said, the technology is often expensive. We can't just bring in when we want. And so we've had to, part of our innovation journey has been to collaborate widely outside. And that's included universities, government organizations like the ARC, other private companies, not just within South Africa, but across the world. And so for us, we excel in what we're good at and what capabilities we have in-house, and then we rely on the strengths and abilities of others and in-source or outsource those things, whether it be sequencing, bioinformatics, particular projects, germplasm. It just makes so much more sense to have an ecosystem of functionality outside of your own expertise.
Thank you. I think this has indeed been very illuminating. I know we're running low on time, but I couldn't just stop them. You will agree with me that this was the best display that one could be subjected to. But we value your participation in this meeting. If I can see by show of hands, those that have questions to ask and comments, just make them very brief. I want to extract as much as I can from the panel. There's a hand from... Okay, Let's take 3 and we'll see where this takes us to.
Thank you very much. This is really interesting. I'm Julius Jalele from the AC. You are said about communal setup. To what extent does the omics mean to a Mrs. Khadebe in Kandla? And again, to what extent does this omics is acceptable by the smallholder farmers in particular?
Thank you.
Thank you very much for the presentations. I'm not sure if you are aware, so within the biosecurity hub, the one aspect is food safety. But what we also, when setting up the hub, so it's plant health, animal health, and food safety, because what you eat and what affects the food we produce also affects the issues of safety and what we put on the table. So my question is, given the challenges we have with food safety, both in the formal and the informal sector, where does one begin to tackle this problem using omics? Where should one start? It becomes one of those big projects, but I don't know if anybody's tackling it with the right tenacity and at the right level. So, open question, because I don't know. Where do you start to unpack what would be a sustainable and a protected food systems in terms of, but particularly using omics to protect our food?
Name and affiliation.
Maneshri Jagmohan Naidu from DSI.
Thank you, Maneshri.
Thank you. Kenny Tenza, CSIR. My question is in relation to the role of omics in precision nutrition. Call it personalized nutrition or call it stratified nutrition. As to whether do you think there is value or there is linkage that one can make between omics and personalizing nutrition. You know, the concept of personalized medicine, etc. So in that same vein, can we then use omics to personalize nutrition? Thanks.
Thank you very much. You got the questions? If we can just go.
So I'll start because I've got a more personal directed. Yeah. Yes. Yeah. Okay. So how I've been using in the communities, I've been using it in a community-based breeding program. The reason why we've been using it as a model or a strategy is that it does not only allow us to do the genomics, but it also allows us to make the decisions at the reproductive level, to choose the animals and also to select for traits. So where omics has been involved in the communities is now we've been doing it for breed purity. So I've got a project in Limpopo where and KZN around in Msinga, where we were testing for purity for nguni. So the whole idea is for them, because they always get left behind in the markets because of the notion that they have crossbreds and. But you'll be surprised that those goats are pure. They are pure ngunis. Some of them were they had higher percentages than the ones that were certified as stud. So what happened at the IVGSA last year at the auction is one of those breeds in the Limpopo, one of the animals in Limpopo by Mr. Modao, they were actually part of the IVG. because they were certified Nguni, even with the support of the genomics. So that's how I've been supporting the farmers. So we've also been doing analysis of the inbreeding and how they can select animals for breeding. So that's how we've been penetrating the communities. In terms of the acceptance, I think the acceptance for us was, the challenge in acceptance was not more for the technologies, it was what the historical relationships that they've had with the companies, or the I don't want to say ARC, but I'll just say Uhulumeni government. You know, so it's us as to change. It's not about the technology. If you were to give them the technology, they are willing, because they know it will improve their market access and guide them. But because of what we've been doing, or the programs that we've been setting up and they don't work, so they have little trust in the organization. So I think the most important thing is for us to fix that. So there's all these technologies we'll be able to implement and be able to penetrate the communities. Thank you.
Just part the question around food safety, and there's also a
Yeah,
question
Manesh's-
around
Manesh's-
precision nutrition.
Yeah. So Manesh's question: how do we get going? How do we start? Well, and the answer is, of course, it starts as a research type project, because you've got to establish what is the right technology to address the appropriate question, and that might be a food identity, species identity question. And then for me, I would say a DNA test is the appropriate approach. But is it going to be a PCR test? Is it going to be a sequencing-based test? Is it going to be a lateral flow device-based test? If you're looking for antibiotics in the food chain, then it's a mass spectroscopy-based test. And so there's a little bit of a question of saying the technology will be appropriate to the question that's being asked. And yes, it's a question of demonstrating the proof of concept, scaling to make it an affordable and scalable test, and deciding on what proportion of the food chain are you going to be able to analyze, because you can't test every steak to make sure it's really beef. But, you know, if 1 in 10 steaks is not a beef, it's some other species, donkey or horse or ostrich or whatever has been substituted. And food substitution is a huge problem globally in the food industry. It's not a little problem that's on the side in South Africa. It's a huge problem. And with it go all sorts of cultural issues. You've got obviously you've got Jewish and Muslim communities that don't eat pork. I come from the UK. In the UK, we do not eat horse. So horse is considered to be completely taboo and utterly revolting, and what on earth would anybody eat horse? But you go to Belgium or France or Germany, they eat horse, they love horse. Horse is Sunday lunch. It's absolutely considered to be a delicacy. And of course in South Africa, there are communities that eat horse and lots of communities that don't eat horse. And so if horse meat gets into the pipeline, but in Europe, the horses that get in the pipeline are rejected racehorses, and they're full of steroids and all sorts of goodies that you don't want your teenage son to get hold of. And if that's the case, you don't want that in the food pipeline. So there are lots of issues, I think, as to why we should be testing, but it absolutely is the case you can't test everything. So it becomes a question of saying, how do we create a system which drives down cost, increases throughput, and ensures appropriate sampling of the food chain, preferably not before you've had a listeria outbreak. But when you've got a listeria outbreak and you're not prepared for it, and you can't prepare for everything that could possibly happen, then you've also got to have the ability to respond quickly before too many people catch listeria, which is apparently rather undesirable.
Okay, there's a question, last question on
Precision. Yeah, omics-based nutrition. I think it can happen at a number of levels. I think the more we understand about our breeding material, if I'm just thinking from a plant breeding perspective, the more we can know the genes that contribute to a specific secondary metabolite, which would be nutritional to someone, let's say tomatoes, beta-carotene or lycopene, etc. Then we're able to combine certain inbred lines together that would maximize that particular trait. And I think added to that is pangenomics, where the more multiple realize actually the carotenoid pathway is far more diverse and complex than we gave it credit for, because we only had line sequence before. Now we have 100. We realize there's lots of other alleles that can contribute a bigger effect to the nutrition of the end product. And so I think Omics has a huge role to play with designer food, if you like. I think in some ways we're designing it already. When you have a salad, you combine multiple different crops together, getting nutrition from multiple different sources. And so I think we're used to the concept. But I think taking it a step further, where techniques like genome engineering or genome editing, you know, with CRISPR-Cas, you can edit 15, 20 targets in one go. Imagine making a tomato disease resistant and more nutritious and, you know, add 5 traits in one single step. I think the possibilities are beyond our conception. So, yeah, definitely there.
Okay, I'm very low on time. I'm 2 minutes after the cutoff point. Jasper, you're itching to say something before I close.
I was itching to say 2 things. The first is that Glendon mentioned that he'd eaten golden kiwi fruit for breakfast, and I just want to reassure you they were not GMO. Absolutely, they are not GMO. They're naturally occurring. They were bred in bred in New Zealand back in the 1960s, actually. And then my second comment would be, is it's very clear from what we've discussed today, sequenced genomes taste better.
Jasper, you couldn't have done better than me. For that to be a closing statement to the audience, let me thank you for your keen interest in the subject under discussion. I think we have learned quite a lot, and our panelists deserves beer or a round of applause. Let me thank them, and in closing say that this was indeed a pleasure for me to facilitate this. I've also learned a lot, but what is clear is that this is going to be business as usual, where we're going to be bombarded with a lot of data. So we need to be sure that we are ready to process this data. It's a lot of fun. But at the end of it, we must always be reminded that there must be impact and meaning in what we do. So thank you very much for being with us.